Santiago Ramón y Cajal was born on May 1, 1852 in Navarre, Spain. His father, a doctor and anatomy teacher, took young Santiago for walks through graveyards to find bones to sketch. His father was trying to inspire Ramón y Cajal, who hoped to be an artist, but was something of a hellraiser. By the age of 11, he had already been asked to leave several schools because of his rebellious attitude, and had been imprisoned for destroying a neighbor's garden with a homemade cannon. The graveyard excursions did the trick. Ramón y Cajal went to medical school at the University of Zaragoza, where his father was employed as an instructor. He successfully completed his studies and went into service in the Spanish army as a medical officer. Ramón y Cajal spent only a few years in the army before coming down with malaria and tuberculosis during an expedition to Cuba. Returning to Spain, Ramón y Cajal continued to follow in his father's footsteps. He pursued his doctorate in medicine in Madrid and became an anatomy professor at the University of Valencia, then the University of Barcelona, and finally at Madrid University. It was during his time in Barcelona that Ramón y Cajal learned of a new development in histology, the investigations into the microscopic structure of tissues. Camillo Golgi, a researcher at Pavia University in Italy, was doing histological studies on the nervous system. At this time, it was not clear what nervous tissue was made of. They could see it was composed of something fibrous, densely packed together, but it was impossible to tell how these fibers were arranged or what the fibers were doing. Some researchers, including Golgi, favored the reticular theory, the idea that the nervous fibers formed a dense, continuous net. Others, including Ramón y Cajal, supported the neuron theory proposed by Heinrich Waldeyer. That is, nervous tissue must be made up of individual cells called neurons. Golgi developed a revolutionary stain made of a weak silver nitrate solution, which completely stained single fibers in the nervous tissue while leaving the surroundings unstained. Santiago Ramón y Cajal began investigating nervous tissue in earnest, making use of Golgi's stain. He would take small slices of brain tissue and then infuse them with the Golgi stain to reveal the underlying structure. He started in the cerebellum and then moved to the spinal cord, and then the cerebrum. Over the course of more than 25 years, he investigated most of the major parts of the brain in humans and many other animals. His talents as an artist were invaluable, as he documented his work with meticulous technical illustrations. Ramón y Cajal's work made clear that the neuronal theory was correct. Nervous tissue was not simply a continuous net of fibers. Rather, it is made up of individual cells. Each one of the cells in nervous tissue, the neurons, consists of a cell body and long fibrous processes which extend away from the cell body, the axon and dendrites. Ramón y Cajal noted that the fibrous processes from one cell appear to make contact with other cells, suggesting a means of transmission of information, and that the entire cell could function as a conductive device. In 1906, Santiago Ramón y Cajal and Camillo Golgi shared the Nobel Prize in neuroanatomy and neurophysiology. The prize was awarded in recognition of their work on the structure of the nervous system. Ramón y Cajal's careful and beautiful illustrations are still used today, and we remember him as the father of modern neuroscience.